So when I was first entering, I found this a bit confusing because I was like, NiPi is this one package. What does this mean? But actually, it is a package, but I think it's really kind of emblematic of this ecosystem that was kickstarted. So NiPi, as you might have guessed, stands for Neuroimaging in Python. And it was originally a Python library for the analysis of structural and functional neuroimaging data. But as you might tell just from that description, that's a pretty broad mandate. And in fact, NiPi really includes many, many different packages now. And I'm going to overview some that are listed, but Satra's going to tell us about way more that have expanded recently. And so kind of now what binds this community of practice together today is the idea that you're using Python to look at neuroimaging data using structural or functional or other kinds of analyses. And you're doing so using Python and in this broad community of practice where you have sort of norms about doing so in an open and collaborative way. Such that the packages within this community, within this ecosystem, all can really build on one another and can enable a lot of the research that's ongoing today in neuroimaging. And some of the affiliated libraries are visible at NiPi.org. I just wanted to show my iteration on the state of NiPi based on those affiliated libraries, just to kind of give you a hint about some of the things that have really been part of this ecosystem, this community for a while now. So many of these, I think, will probably look very familiar. Things like DuPre, which you've already heard about, NiPipe, which we just heard mentioned, NiBabble, NiLearn, M&E, PyMVPA, lots and lots of things that are all kind of building on these ideals of open source development in Python in a collaborative modular way to enable structural functional neuroimaging analysis across different scales and different modalities. Obviously, though, this is not the entirety of what would be considered broadly under the NiPi umbrella. And so I think that's one of the things that I'd really like to talk about today is kind of what's the state right now, even beyond just the website in terms of what we think about as part of NiPi. I'm going to turn to Satra to talk about that a bit more. But one thing I did kind of want to plug here is that one of the nice things about having this as a community rather than a monolithic stack is that this is something that's constantly growing and evolving. And that's sort of a double-edged sword, right? In that, like, on the one hand, there's always something new. There's always somewhere to jump in. On the other, there's always something changing. And so trying to stay abreast of what's going on and connected isn't straightforward necessarily, particularly if you're coming in, you don't have guideposts for where to look. So I just wanted to highlight two different venues, which I think are really, really useful for this. The first is the NiPi developer discourse. So that's what I've taken a screenshot of here. So this is relatively new, and this is a forum that's specifically designed to make it so that if you're developing something within neuroimaging in Python, broadly construed, and you'd like some help to think about, how does this lock in with other pieces of the ecosystem? How can we work together? This is a great forum to do so. If you're not developing on a particular project, but you have a question, there's also another really great forum, which is NeuroStars, which is partially sponsored by INCF, the International Neuroinformatics Coordinating Facility. And that has a really amazing, at this point, it's been running for a few years. And so it's got a depth of resources of previously asked questions that you can kind of look through and see if they can help for your specific use case. And of course, many of the developers hang out there and are happy to answer questions all the time about their own tools. So that's kind of the 30,000 foot view of what NiPi is historically and what it sets out to be. But I think, Satra, you have some more specific points about some packages. And I'm happy to just click through the slides.
Wonderful. Thanks, Elizabeth. So I just wanted to kind of touch on a couple of things, both based on what you may have seen in the videos that were part of kind of a pre-watch party before this session. And one of them is kind of what's happening with NiPi. And one of the things I would like to say is NiPi, the current NiPi, is a very mature package with lots of contributors, and it's being used for a lot of things. But that said, software and other ecosystems evolve around it, and even some of the things that it was originally created to do, it cannot do other things without kind of some structural or functional changes to how we implemented the architecture. So, towards that in mind, and the fact that a lot of the scientific ecosystem is living in Python, we turned towards creating an underlying kind of workflow engine called Pydra, which will form the basis of this new NiPipe 2.0 ecosystem. It's still it's being used in a bunch of projects in our group, but it's something worth kind of looking into and thinking about as you simply even write scripts. And that was one of the biggest changes from NiPipe to Pydra, is that we should be able to turn your Python functions, if you're writing Python, into a workflow system very, very quickly. And that's the kind of thing that Pydra does. But Pydra does not sit alone. There's another framework called TestKraken. As you may have seen in the world of neuroimaging, more and more studies are coming out that are suggesting there's a lot of variation across workflows and choices of parameters that people make. And so TestKraken was created with the intent that you can compare things and figure out what the sources of variance or noise are in workflows. Sometimes they come from the algorithms you're using, and sometimes they might come from operating system level things. I don't think Greg is going to be talking at NeuroHackademy this year, but Greg Kier, a recently minted PhD, did his PhD on trying to figure out how math libraries in various operating systems influence the scientific output of things far away from those math libraries. And so, things to kind of consider as you think about science is, how are you modeling the system that you're observing? And how does that map on to these tools and resources that you're using? And so TestKraken was an effort towards that. NeuroDocker kind of allows you to create Docker images or singularity images for kind of customizing analysis environments. If you start installing all the imaging tools into a Docker container, that container grows big really, really quickly. So you may want to kind of separate out some of the things that you, if you're only interested in sort of, let's say, NiBabel and some of the Python tools, you might not need a Docker container. A Conda environment might suit you just fine. But if you're looking at other things like FSL or SPM or other things, being able to kind of pull them together into a consistent environment is what NeuroDocker provides. And then NiFlows was kind of an abstraction on all of the workflows that have been created, but with the notion that it's really hard to find what's out there in terms of workflows and to know how they have been validated. So this is a very early project, and it builds on things like NiPreps and other things that are in the picture, and I'll talk about NiPreps in a second, to say there are neuroimaging workflows. Not all of them are in Python, but it would be nice if we knew where to find them. Right now, a lot of things are by word of mouth or by reading a paper or seeing something. So could we create kind of a registry that brings together in some consistent manner the set of workflows? And that was the intent of NiFlows. We're hoping, I mean, that's a big part of a future cycle of a grant that's coming up, so that will come into view. But I wanted to give you at least the notion that this will come to exist, and for those of you who are building workflows, think of it as kind of the model zoo equivalent for workflows. And if you're involved in genomics or bioinformatics work, you have already seen some of this in something called DocStore, which is a wonderful place to find various kinds of workflows for doing genomics research. This is really kind of the extension of NiPipe 2.0. Next slide, please, Elizabeth. And this is where the fun begins. If we look on the left, when NiPi started, all of those things did not exist. And that's the scale of national and international databases and archives that we now have access to. Things that we can pull even before deciding that, I need to collect data myself. You can now reach out to many of these sources to find data that might be relevant to you. Yes, some of them are open, fully open, like OpenNeuro, and some of them require restricted access, like the ABCD dataset, but you can get access to it. The UK Biobank also falls into that same thing, The Child Mind Institute's Healthy Brain Network is a mix of open and restricted data. So you can get open data easily. And if you needed more phenotypic information, you can get to it. The Human Connectome Project, very similar, a mix of open and restricted data that you can get access to. But all of these are accessible, and that's the key point over here. And that means you can go across multimodal domains that interact across various spheres of neuroscience. So that's on the left. On the right, within that brain outline, is a set of things that have been built on top of these tools like NiveAble and NiPi, the bid standard, and PyBids. And this forms this NiPreps ecosystem, and which I think as of just 2 days back received the formal award announcement for the next 3 years as a Brain Initiative software project. And Ariel is one of the co-PIs on the project. So if you have questions about NiPreps, you can reach out to Ariel. But I think it involves a team from Ariel, Russ, Oscar, Ted, and Mike Millham.
That's correct. That all
There's a wonderful collection of people kind of working towards improving pre-processing stages, which are so fundamental to a lot of the things you do in neuroimaging. And this is also built on some of the pieces like DataLad, a tool that you may have heard of, or if you have not, you should hear of it, that provides access to a lot of these data that are sitting on the left, and something you want to kind of pull into your research workflow if you haven't already. And I'll make a slight mention of No-Brainer, which is a separate project that we are running. So this is, again, a view of how this NiPi ecosystem is growing. So you may have noticed there are machine learning tools, there are pre-processing tools, and other things that Elizabeth mentioned earlier on. The world around us is doing all kinds of deep learning. So No-Brainer is a project that we have been running now for about a year to try and consolidate that deep learning space towards not computational models of brain function. So there's a world of neural networks that's focused on how do neural network models map onto human behavior or do things like a human. The intent of No-Brainer is not that computational neuroscience space. The intent is kind of the production engineering space, that there are lots of tools that are there that we need to run very, very quickly, especially given the sizes of these data sets that are now online. And so No-Brainer is intended, and if I could go to the next slide, to create baseline networks so that everybody does not need to retrain their models from the scratch every single time. That's a lot of GPU computing and electrons being used. So if we can kind of create a core base of baseline models, you can pick up on those things. We're also interested in distributed computing. As the world evolves, we believe that not everything will be open. There'll be a lot of data behind restricted sources, so we need to be able to compute on those data as well. And so we're looking at privacy and interpretability in the context of this mixture of open and closed worlds. And finally, just like many of these other tools, a lot of software engineering just to make things simpler for people. Can you just download a model and run it towards inference? So we hope to use and leverage some of the same data sets that I showed on the previous slide and create tools and algorithms that kind of minimize or speed up a lot of the basic tasks. And hopefully some of these will get into the NiPreps framework itself as some of the underlying pieces of some of those workflows. And I'll end, I think, with the last slide, which just says, part of the goal for us is we are in a lucky position to have access to some incredible resources to train neural networks. Not everybody has access to those things. So we welcome community networks and models to be placed before us and say, no, dear No-Brainer team, can you retrain this network on all the data that you have access to and create a model? We want to be able to do that. So if you have models that you would like to see retrained on larger data sets, because we want to address bias and variance that exists in a lot of the data and make it clear when the model can work and when it fails. And so please reach out to us. There's a code base already. So I would say in some ways No-Brainer might be the youngest library in the NiPi community of practice ecosystem. Maybe there are more younger ones as well. But we are looking for people to contribute models to it, but also reuse the models we are generating. And if you have models similar to what we were talking about in NI workflows or NI flows, we are creating a model zoo. The idea there is there are many people producing neural network models out there. Again, hard to find, hard to validate, hard to know what they are capable of doing. So we want to leverage some of the things we have done over the years in the NiPi sphere of consolidating efforts and bringing to everyone a set of things that they can look at. I think that's it. So I'll stop there. And anything around these things, I think Elizabeth and I would be happy to kind of chat, take questions. We really wanted this to be an interactive session.
Yeah, so let's see, what questions do people have? That was a lot of different things, and we can pull the thread out of that to go in different directions here. What people have questions about? Happy.
Hi, I just wanted to say this is actually wonderful. So I'm going into my second year in my PhD, and of course, I'm recently venturing out into learning about FSL and SPM and AFNI, and it always seems like a war between these three, and I feel so conflicted as to which one to use at the end of the day. So if I understand it correctly, NiPi and its connecting libraries can help me consolidate those three into one environment. Is that correct?
So I can take that on. So NiPipe could allow you to mix and match tools from those three packages into a single environment. I think there's a more fundamental thing to your question that I'll pull out, which is your first statement that you made, which is you see these three as competing forces in some ways. And that might have been the case. I mean, one is in MATLAB, right? One was a completely open tool that could be used by anybody. The other was an open tool, but could not be used for commercial applications without a license. And they had kind of different mindsets and variances as to what they were targeting when they came out with. And I think that's where the richness of those tools lie, which is they adopted different models for doing the analysis. So there's some wonderful work by Camille Maume and Tom Nichols and others showing how if you are doing functional imaging analysis, fMRI, that the error models that are implemented by these tools are different. And that goes to a more fundamental question about which model should I use? And I don't think the neuroimaging world has come to a consensus on what should be, but that gives you an opportunity to see, is your scientific result generalizable across those error models or tools? So I would see it through the lens of kind of scientific questions rather than competing toolboxes. That being said, it does add to the mental bandwidth required to know all the tools that exist in the ecosystem.
Thank you. I'm pretty sure I'll have follow-up to that, but I'm going to stick with this.
Yeah, Priya, I see your hand is up.
Hi. First, I just want to say hi to Satra. Hope you're doing well.
Good to see you.
Yeah. Keep popping up in strange places. And I was just wondering, so if somebody's just going to start learning to use NiPipe, do you recommend, like, is the Hydra version all up and running to just start there?
That depends on what you're doing. If you're in kind of already familiar with Python and writing Python functions, then I think Hydra is at a place where you can easily launch off of it. When it comes to the neuroimaging tools, there's a lot of work going on in building out the corresponding kind of Hydra tasks to the NiPipe ecosystem. So there I would strongly say, if you're interested in neuroimaging tools, stick with NiPipe for now while that comes online. But if you're in the Pythonic space using DuPre or NiLearn or other things that are kind of locally parallelizable, but when you're looking at scales of 20,000 or 30,000 or 60,000, that's where something like Hydra would allow you to easily attach some of those things to your computational clusters, as an example. And there are a few other tools. Clouder is another one that you can use to do some of those things. So I would say, think about your use cases and where you want to see. Also, I mean, as you just heard, NiPreps is this newly awarded thing, but while it's newly awarded, it has been going on for a while. If you want to contribute to that, almost all the tools there are built on NiPipe. So NiPipe would be a good place to start if you're going to be contributing to that ecosystem.
Great, thank you.
And as part of that contribution, I'm sure parts of that ecosystem will migrate to Pydra, and you'll get your exposure towards that.
So building on what Hetty said before, would you say that SMRI prep and its equivalents would help us maybe resolve the tension between all those different packages and come up with a completely robust, standardized pipeline that we can all use, and or
Hi, Z.
we're forced to mix and match.
Hi, Zvi. Good to see you again.
Yeah, great to see you. Glad to be here.
I don't know about the kind of underlying social answer to that, but what I can say is that I think you should use the tools that are needed for the job at hand, right? And the example I'll give is spanner wrenches, tires, the kinds of things that you end up using. You know, if you want to go at 160 miles per hour, you're not going to buy a tire that wears out at 88 or 90 miles per hour. If you take a spanner wrench that's supposed to open a 10 millimeter bolt, you know, if you take something that either does not fit or is too large, it's not going to work. And I think a lot of these things come back to asking that question: what am I using this for?
And in a sense, I guess I'm asking, what do you think is the limit of our ability to abstract these commonplace procedures that all of us have to do? We need to pre-process our data, and there are so many different levels of variation to that. So, you know, there's an 80% that's valid for.
And I think, well, I'll take two pipelines, at least for fMRI, that are out there, right? The HCP pipelines, if you are collecting that kind of data, fMRI prep, right? And they do some things similarly and some things differently. I do think that one of the things that both are doing is trying to consolidate knowledge in the field. To say, here are some of the better practices of doing analysis. And I think that that's a part where it's still fluid. We are not at a stage where it's a production system where we understand all the pieces of knowledge. And so the effort there is to say, okay, everybody does not need to understand all the pieces of knowledge. Here, as communities of practice, we've put forward these things, right? And I know Mike is not here, Mike Millam's group, but CPAC, right, provides another way of building out these workflows and pipelines to optimize towards your use case. So I do want us to think slightly differently. Yes, there might be a consolidated pre-processing pipeline, but your use cases are likely to change over time, even for something that might be seen as standard as pre-processing. Because scanners are changing, sequences are changing, your target audiences, not target participants are changing. Hopefully target audiences don't have a role to play in the pre-processing workflow. But so I don't know.
Assuming human, sorry.
So I don't know if there's a good answer to that part of the question. I think it's going to continue to evolve because that's what science is, and we're still not at that stage.
Assuming human judgment is suboptimal, just, you know, throwing it out there. Would you say neural networks, in that sense, might replace a lot of those decisions? So if your images have different qualities now?
No, I mean, we're not there yet. I mean, if you think that our pre-processing, which we've been doing for 25 to 30 years, is still not yet set in stone, the other thing is, I would say in that horizon, just an eye blink relative to staring at a page for a while.
Hi, I see your hand is up.
Yes. Hi, Satra. Great presentation. This is a great tool. And I was wondering, is there something similar to Combat? I know that Combat is currently implemented in several languages, one of which is Python. And if I was going to be harmonizing different data sets and then using NiPipe, is there something like Combat already in the workflow that, or is there a way to implement something like Combat into the workflow, into a workflow that could help harmonize different data sets?
I don't know the answer to that. Elizabeth, do you have a...
I know Dylan Nielsen was working on something like that, but I don't know the status of it right now. I think one question might be just if you process, it might make more sense, my gut, but I do want to look and see what Dylan and others have done, would be to do the processing for each data set separately and then take whatever summary stats you're trying to derive and then use Combat on something like those, rather than trying to integrate different site information directly into the processing. Because that goes back to kind of what Satra was saying before about like the processing does in many ways interact with, you know, the different sites, like we have different acquisitions and those acquisitions will require slightly different choices. So disentangling the site effects from the processing effects at that stage would be difficult, but maybe once you have some kind of estimates to work with, it might be a bit easier.
That's how I'm currently using Combat at the moment, is on all the estimates that were collected on different scanners. But I don't know if we know yet. Like, we know that's how Combat is generally used, but I don't know if it would be like efficient in a different way if it were to be implemented prior to that. But I don't know. Is that not? I guess that hasn't been done.
As far as I know, I haven't seen an instance of that at an earlier stage. And I believe all of Dylan's work is also at that post-analysis stage of aggregating things. The part there is some work is not through Combat, but through harmonization tools and algorithms that try to look at the data directly out of the scanner and try to say, how can I process the data before it even goes through pre-processing in some ways, right, to get at kind of equivalence. And a lot of this is happening as part of the ABCD study, which is a multi-site acquisition, and the forthcoming HBCD study, which gets to a 0 to 3 year range of data collection, where there are other kind of issues that come up in that kind of data. And there's been some good work out of UNC, Chapel Hill, and others in doing some of these image level harmonization tools. And I believe DuPre has a set of tools, and Ariel might be able to speak to some of those pieces, kind of a little bit of denoising or other things that happen pretty much immediately after things come out of the scan.
And Elsteris presented some of that yesterday when he spoke. So,
wonderful.
So, yeah, it's kind of patch to self. And I'll post another paper from Daniel Moyer and colleagues about the harmonization stuff that happens at the first stage. But yeah, just to add to this discussion about Combat, is that one of the implementations that does exist is in the form of a scikit-learn compatible estimator. And those of you who hear about scikit-learn will hear what that means. And that means that you can plug it into inference pipelines of that sort, including everything that is in NiLearn. You can put that in as part of your pipeline. And what's nice about that, but that's at the inference stage, right? We've derived the features already. But then everything kind of works. Pretty automatic. Very nice.
Can I ask one more question about imaging? This is, since you both work with many workflows and doing pipelines, what's your opinion? We're working with some multi-band data and fMRI data in our lab on people who have bipolar disorder, and it's been analyzed in a couple different ways, one of which is ICA and one of which was fMRI prep. And I know there's different benefits to both, but one of the most important things is the motion artifacts and trying to save movers. What are both of your opinions on ICA versus fMRI prep?
Sorry, can I just ask a clarification? So fMRI prep, when you say ICA, I think you're doing something like ICA aroma, is that right?
Yes, yeah, to denoise the data.
To denoise the data, and fMRI prep does include ICA aroma, optionally. So I assume you're saying like with or without ICA aroma, is that right?
Yes, that's a better, more clear way to phrase it.
No, perfect. I mean, Satra, I don't know if you want to jump in. I would just say that it really sort of depends on what you're trying to do in part, right? So ICA aroma, what it's basically doing is it's doing a decomposition, and then it has a series of rules that it applies to those components and tries to say, do I think that this is signal or do I think that this is noise? And so for some data sets, those rules work very well. In fact, probably for the majority of data sets, which is why ICA aroma has been so successful. There are other data sets, however, particularly some really high multiband factors or like precise acquisitions where those rules don't work as well, and they can break down, and you can see that things that are actually noise are labeled as signal and vice versa. And so I would need to know more about kind of exactly what you're doing, but I think that just in general, you know, there's never a one size fits all answer. But the approach that you're taking with ICA aroma versus, you know, if you're not doing ICA aroma, you might be doing something like scrubbing, you might be doing something like including the motion regressors. There's lots of variability in how you're looking at it, but that's kind of what I see as the fundamental idea with ICA aroma is just, if I do a decomposition and I apply these rules, how well do they describe or delineate signal versus noise in my data set based on what I expect from other data sets?
Thank you.
Yeah, I would just to jump in here, I would just add to that also that you want to define an objective measure to say, and one nice thing about all these tools is they let you run these things head to head and compare. Did I turn on this button or did I not turn on this knob? And then, but you need an objective measure that would tell you temporal SNR, the ability to detect a certain activation due to a task, something of that sort that would serve as a criterion for saying which of these choices is a better choice for this case.
Yeah.
And it gets pretty hairy.
Can I follow up on Ariel's point a little bit, because he mentioned temporal SNR, which is one of my favorite things. What would happen if you took your time series and just smoothed the hell out of it temporarily? What would happen to TSNR?
You asking me?
I'm asking generally, anyone.
I'm not as familiar with like what
Here.
that would be yet.
So temporal SNR is defined, so I'll give it to is mean of some signal over the variance of that signal. Right. So if you could reduce the variance of that signal, what would happen to the number? It would go through the roof, right? And so my ideal TSNR would have a constant through time. So one of the things, and this is fun because people will say, how's your TSNR? And I think it's contextualized in a lot of places as to where you're looking for TSNR. Because if you tell a machine, optimize over my TSNR, it would try the fastest possible route to turn your signal into a constant over time, which would be exactly the opposite of what we might be looking for in terms of an optimization. But speaking to that point that Ariel brought up, I think as a community, we need to understand what we are optimizing towards. How would you compare those outputs? Often we see it through the lens of, are my expectations of where I expect to see patterns of activity met? I don't think that has changed a whole lot in the last 20 years. But are there other ways of thinking about it through predictive models, for example? That you can build a model that says, I can predict activity in a new subject I have not seen because my model is good enough, right? And now
you could compare like how well they generalize or their like accuracies, how well they are above chance. I mean, ultimately, we're trying to move towards being able to replicate other people's findings. And I guess with all these different pipelines, we would want to compare, if we have the same end goal, we'd want to compare the pipelines into how well can we replicate each other's findings.
And a notion for what you mean by replication. So I think it would be good for you to decide, this is my objective criteria for what I mean a good replication should be.
Right.
Elizabeth, sorry I cut you off earlier.
No. I was just going to kind of plus one your point. I think that one thing to be careful of too is that it's very easy to trick yourself if you're looking at your data set and you're running many, many different pipelines on the whole data set and then sub-selecting from those, right? In the same way that, you know, if you're developing a predictive model, if you train and test on the whole data set that you want to work with, you can sort of delude yourself and think that like, oh, I'm doing really well, when in fact you're just overfitting to the particular idiosyncrasies of your data. So if you are going to use your data to kind of explore this pipeline space, that would probably be something you'd want to decide ahead of time, maybe with a pilot or with, you know, a sub-selection of the subjects that you have access to, and make that decision independently from running it on the entire set. Because I think otherwise you can end up in a situation where you've maybe overfit or overdescribed your particular data set rather than trying to provide something more robust.
Yeah, one thing, I'll wrap this up. I'm almost done. I was just gonna say one thing we plan to do to mitigate, to help with something like that is to have like a small replication sample that we would use that is some of the same subjects, but they were scanned on a different scanner. Anyway, thank you for answering all of my questions. I didn't mean to totally monopolize, but this was really helpful. Thank you.
Hedvi, I see you have your hand up. Do you want to ask a question?
Hi. So this one is a little bit more technical. So I was wondering how mature the PyMVPA library packages so far, and whether there are any packages, including the PyMVP or anything else, that can even help with the graph theory.
So maybe Satra and Ariel, I'm sure you have great context here because PyMVPA has been around for a little while. I would say that PyMVPA is very mature, so much so that it's not under super active development right now. It is maintained and it still has access to everything that's been built up over a long period of time. Like, for example, if you're interested in the hyperalignment algorithm, this is kind of the core library to have access to that. And so that's available there. That being said, so it is very mature. I would not think of it immediately for graph theory analyses. I would think of something like Network X maybe for graph theory analyses instead. So PyMVPA does have things like MVPA proper, so multivoxel pattern analysis and hyperalignment. If you're interested in things kind of in that vein, there's also the package from the Princeton Neuroscience Institute, BrainIAC, which implements those things and adds in more recent developments, for example, like the shared response model. But yeah, for graph theory, I'm first like thinking of Network X. I don't know if folks have other recommendations as well.
Yeah, I think it also, we could generalize this a little bit. There's sort of, I think when you look at the explosion of software that exists, you might ask, how do I find the software to do X? How do I know that that software is software this is the software I should be using? Let's say I'm asking myself, oh, how do I do graph theory analysis of brain data using Python? You know, my first thing is to Google that and see, and then so the question is, now I have like 5 different options. How do I look at that and select among those options? What's your thinking?
I don't know what the state of it is. There was something called the connectome mapper in the middle part of the last decade that was exactly trying to say, okay, Network X does a lot of things at the low level of extracting various kinds of graph things. Connectome mapper was combining that together with neuroimaging, but I think that project may have died like many software projects do, but I'm not completely sure. If it's
so to answer my own question a little bit, what I do is I say, hey, Satra told me about connectome mapper. Let me go and search connectome mapper. I heard about this. There was someone called Stefan Gerhard who developed the software, but that was like 10 years ago. Is that still active? And so then I Google the thing. I find the link to GitHub. Sorry, I'll share that. And I look at it and I say, well, how active is it? Well, one thing to say is somebody committed something here on March 7th. It's not that long ago. Then you might ask, okay, who are these people? When, so you look at this and you say, oh, 5 months ago, 7 months ago, 9 months ago. that's one metric, is how active is the project itself. Seems not too bad, actually. And then you might look at, the documentation and see, well, how much documentation is there here for the particular task that I want to do? Well, what does it do? Does it do the thing that I want it to do? This one's pretty well documented. This project has been around for a while. And then how do I use it? Is this usage appropriate for what I'm trying to do? And so on. That's sort of one set of strategy. But yeah, looking to see how active a project is, as when did people recently do anything with this project, is pretty, to me, is a metric that I look at. Turns out Connectome Mapper is still alive.
So Stefan is no longer in the picture, but Sebastian, he has been active in a couple of different circles in the BID/NiPi world of things. Yeah.
And maybe to kind of follow up on Ariel, your point about like how to see if packages are active, or like how to think about what package to use. So I do think like ongoing development is a big thing. One thing you do have to kind of look for there is like, though, it sort of depends on your goals, right? Like if you're designing a software yourself, or you're designing a pipeline yourself, and you want it to be able to work over time, and you're thinking about what dependencies to plug in, you want to think about, can I rely on this over some period of time? If you're doing a one-off analysis and you're just going to containerize it, and you're like, here it is, it's done, it's on my shelf, and you can pull it and replicate my paper, like that's a slightly different situation. But let's say you're in the situation where you'd like to be able to work with a library for a period of time. I think looking to see how active it is really important. Also looking to see kind of how it's run. Is it one person? That's a huge question, because, we all get busy. I'm trying to do my dissertation right now, like I totally admit that I'm falling off the earth on some things. And so like, it really depends on a particular person. Like if it's down to one person, they're going to be a bottleneck whether they want to or not. I think you may have heard this already. There's like the idea of the bus factor in open source. I think the really pleasant way to think about it is the bus that picks people up and takes them to work at Google. So like if there's only one person working on your project, when the Google bus comes for them, where will the project be? Versus if you have a community of people working on the project, then you're in a slightly more sustainable situation where you can work with it over time. And it also makes it easier for you to jump on board and hopefully be able to contribute developments and kind of help take the project in a place where it's really useful for you and the kind of work you're doing. So that's the other thing I'd sort of plug in terms of thinking about projects to use over the long term. But that's not to say that if a project doesn't meet those criteria that you shouldn't use it. It's just a slightly different way to think about, at what time scale you're going to be using it.
Okay, great.